I'm Kieran Mulroney. I'm a PhD candidate at the Harry Perkins Institute of Medical Research, which is attached to the University of Western Australia. My research focuses on rapid diagnostics in severe bacterial infections, specifically looking at the antibiotic resistance profile of those bacteria that we isolate. When a patient has a severe infection, using the correct antibiotic is very, very essential in terms of whether or not that patient will survive the infection. Antibiotic resistance is rapidly increasing within the community, and it's a case in which bacteria are no longer susceptible to the commonly used drugs. So we need to be able to identify that very rapidly. We've been using flow cytometry to speed up the process. The flow cytometer, which uses a series of lasers to get information about the bacterial cells, to look at the drug's effect on individual bacterial cells. We use the Attune NxT, which is an acoustic focusing flow cytometer from Thermo Fisher Scientific. We've chosen this one because it has a particularly strong resolution on small particles and being used to rapidly analyze samples either in pure culture or directly from clinical samples. This instrument has a couple of really useful features for us. First of all, it is very small on the benchtop, which allows it to be put into a smaller lab without use of core facilities. It has a wide range of configurations that allow us to study as many markers as we wish to, and it also has specialized fluidics that allows us to do accurate volumetrics of the particles that we're measuring. Accuracy becomes important when you're looking at single-cell technologies like this. So for us, we're looking for the effects of the drug on individual cells in a very rapid manner. So without the level of precision we can achieve with this instrument, it becomes impossible to measure single-cell responses to the drug. Traditional antimicrobial sensitivity testing can take between 1 and 3 days, depending on how difficult the bacteria is to grow, isolate, and then test for sensitivity. However, we can, using the flow cytometer, do this much more rapidly. Within 1 hour, we can tell you whether or not the drug will be effective, so susceptible or non-susceptible, and within 3 hours, we can give you a full characterization of inhibitory concentrations. Moving into the future, we hope to move our technology into the parts of the world that need it most. So moving towards microfluidics and deployment into the developing world, such as Africa, Asia, and the poorer parts of South America. We aim eventually to characterize a full suite of drugs and bacterial combinations that will allow us to move this into a clinical lab as soon as possible. The technology required to accelerate development of these sorts of tests will either be advances in flow cytometry, whether that is new detection methods or bespoke hardware for specifically testing in this manner, or moving into a microfluidic space that can be more readily field portable and grant access to those areas of the world that need it most.